Hello students, this is Inheritance and Variation Part 6.
Now, what is a dihybrid cross? So, in a dihybrid cross, a cross between two pure homozygous parents. So, I have explained earlier what is a pure trait, what is homozygous. We have talked just. Okay. So, a cross means when we breed two pure homozygous parents in which the inheritance pattern of the two parents in which the inheritance pattern of the two pairs of contrasting characters is considered. So here we consider two different parents and the two different contrasting pairs of the characters. Okay, not only a single character, we not Mendel just not considered a single character, that is stem of height, but he also considered the color of seed. So stem of height and color of seed. Or if you want to consider a different example, suppose he concentrated on the shape of seed and color of seed. Okay, so at a time two pairs of contrasting characters are considered simultaneously, so that type of cross is known as dihybrid cross. So let's move to further. Dihybrid, it is heterozygous for two traits. It can be heterozygous, okay, hetero and homo, okay. It is heterozygous and homozygous. Now this is heterozygous. Two traits are produced in a cross between two pure parents, differing in two pairs of contrasting characters. So dihybrid cross, we consider the two pairs of contrasting characters at a time. So in the next slide, dihybrid cross in that experiment, what Mendel did, the same experiment, the same steps he followed, which he followed in the monohybrid cross. Okay, so these three steps are: he obtained pure lines by selfing the parent plants for 3 generations. So what is selfing? Okay, selfing is if we cross the two plants of the same generations, F1 generations, the plants which we get in F1 generation, if we cross the same plants in the from the first generation, it is selfing. Okay, and if we move further, that in the second generation, in the F2 generation, if we cross the plants of the same generation, that is in the F2 generations, plant of F2 generation, one plant, and again it is crossed with the another plant of the same generation, F2 generation, then that is known as selfing. Okay, selfing of the parents. Now what is pure line? Pure line, it is the particular genotype. It is when self crossed for 3 to 5 generation. When it is self crossed, then we get a pure line. Okay, and what is the purpose of pure line? Pure line is the purpose is to obtain the same type of genotype. Okay, capital T, capital T, now it's a same type of genotype, and small t, it's a same type of genotype. Okay, so when we want the same type of genotype, then we have to self cross the plant for 3 to 5 generations again. Okay, so then we will get a pure line by selfing the parent plants for the 3 generations. What is the next step? He performed artificial cross by using the technique called emasculation. So we have already discussed emasculation in very detail by considering the structure of the flower of pea plant. In the emasculation, male pollen grains, they are cut by using artificial method, by using scissors or by using forceps. Okay, and by this emasculation process, the artificial cross was carried out. Okay, so if you want to understand what is artificial cross or emasculation in detail, just listen the previous lecture. So the third step which Mendel carried out, that he sowed the seeds and raised the plants of first generation and allowed the selfing to obtain the second generation. So this was the third step which was carried out by Mendel. So what he observed, that the new combinations of the characters in the F2 generation were found to be present in case of dihybrid cross. Okay, and he established the law of independent assortment, where he found four types of gametes due to segregation and independent assortment. Okay, so this point is new, and this point is it makes the dihybrid cross different than the monohybrid cross. So you can observe this point, that how the four types of gametes newly formed by a process that is known as segregation or by independent assortment. Okay, so that you will come to know in this by studying this diagram. So if you are observing the diagram, see, this is the parental generation. Okay, this yellow round seed. Okay, observe the shape of the seed. He considered two characters. Di means two characters at a time. Okay, so he considered the shape of the seed. This seed is round while this seed is wrinkled. Okay, this seed is yellow in color while the other seed is wrinkled. Okay, so this is a parent generation. What it did, he observed the phenotype. Phenotype, as I have explained, phenotypically these two seeds are different. Okay, what is genotype? The yellow round seed, it contains all dominant characters capital Y, capital Y, capital R, capital R. While this green wrinkle, it contains small y and small r, small r. Capital Y, it stands for the yellow color, and capital R, it stands for the round shape. Okay, small y, its recessive character, it represents the green color. Small r, it carries the information for the wrinkled type of seed. Okay, so the gametes which are formed by this yellow round plant, all gametes are same, capital Y, capital R, and in the second, small y and small r. In the first generation, that is in the first filial generation, by crossing these two characters, what results Mendel observed, that all the plants of the first filial generation, they were carrying all yellow round seeds. Okay, so all the seeds, they were yellow and round in case of dihybrid ratio, and all of them, they were carrying capital Y, capital R, small r type of gamete. So it is these are homozygous traits and it is a heterozygous trait. Okay, so the gametes are mixed here if you are observing. So when there is separation of the gametes, when the selfing of the F1 was done, so what Mendel did in his next step, that he self crossed the same plants of F1 generation. When F1 from the first generation, he self crossed the two types of plants. Then selfing, when it was done, then he got four types of gametes. Okay, the gametes, they got segregated, they got separated, capital Y, capital Y. Okay, now this capital Y, it can also get combined with this capital Y, small r, capital Y, capital R, capital Y, small r. Okay, now this small y, it can get combined with the capital R, and this capital small y, it can also get combined with the small r. Okay, so these are the four types of gametes which independently assorted, independently expressed themselves by a process of segregation, and this is known as independent assortment. So by selfing the dihybrids, what Mendel got in the F2 generation, he used Punnet's square. Okay, so we have talked about Punnet's square earlier. In the Punnett square, we write the gametes produced by the plants, and the boxes give us the probable numbers of the phenotypes or the genotypes in the respective generation. So in the Punnett square, we take into consideration the male gametes and the female gametes. Okay, male and female gametes. So we capital Y, capital R, capital Y, capital R. These are the combinations. Okay, so small y, small r, small y capital R, and small y small r. So these are the combinations which he got in the F2 generation, that is in the second filial generation. So those characters which are not which were not present in the parental generation. For example, Mendel started with the yellow round and the green wrinkled seed. Okay, but surprisingly, in the F2 generation, Mendel found that some yellow seeds were wrinkled. Okay, so a wrinkle character was in the dove green seeds. Okay, so that character was found to be present, or it was expressed in the F2 generation. It is a new character. Okay, and some green seeds. Now these green seeds, all the green seeds are wrinkled, but in the F2 generation we found some green seeds which were round. So there were variations in the shape of the seeds, there were variations in the color of seed, and again new combinations he observed in the second filial generation. So what Mendel stated that because of the segregation of the gametes in the F1 generation, when he self-crossed the F1 generation and he obtained the F2 generation, so due to segregation of the gametes and due to their independent assortment, due to their independent expressions, he the law of second law of dihybrid cross is completely different than the monohybrid cross. So the ratios are again different. In case of monohybrid ratio, if you remember, the phenotypic ratio was 3:1 and the genotypic ratio was again completely different. The genotypic ratio in the monohybrid was 1:2:5. While in case of dihybrid cross, what Mendel observed that the genotypic ratio is 9:3:3:1. Okay, while phenotypic ratio, while the genotypic ratio is 1:2:2:4, 1:2:1, 2:1. Okay, so these are the number of different combinations or new combinations which Mendel observed in the F2 generation. This is phenotypic ratio and this is genotypic ratio. So these are different variations which Mendel observed in the law of segregation or law of independent assortment. So what exactly, after performing all this experiment, what exactly Mendel proposed in the dihybrid cross or in the law of independent assortment, that we will see. So in the law of independent assortment, this law, that is law of independent assortment, is also known as third law of inheritance. Here, when the two homozygous parents differing in two pairs of contrasting traits are crossed, okay, homozygous parents, if you remember the earlier slide, capital Y, capital Y, capital R, it one is one homozygous trait. When it is crossed with that parent, when it is crossed with the another homozygous trait, that is another homozygous plant that which carries small y, small r type of genotype. Okay, so genotype genes are same, so that those plants, two plants are homozygous, but what they differ in two pairs of contrasting traits. When they are crossed, the inheritance of one pair is independent of the other. Yellow round and green wrinkled. So these two characters, so the inheritance of one pair, yellow round pair, is independent to the green wrinkled type of seed. Okay, so that is when a dihybrid forms gametes. If you remember the earlier slide, the gamete formation, while in case of that, the assortment or distribution of alleles of different traits is independent of their original combinations in the parent. So there are different types of distribution of alleles were found by the selfing of F1 generation, and which were observable in the form of observable characters, that is in the form of phenotypic character. Mendel observed it and he reported that there is independent assortment of the alleles, or there is independently the alleles, they are distributed. Okay, and so this is the law of independent assortment. So what is the difference between a monohybrid and a dihybrid cross? So here we are going to again observe the same images. This plant, consider this is again one plant, that is Pisum sativum, and the same plant is observed in the next image also. The difference is in this one, that is in the monohybrid cross, we talk about the one character, that is stem height of the plant. Okay, while in case of dihybrid, here round yellow pea seeds. So here the color of the seed and shape of the seed, both the characters are considered. While in case of monohybrid cross, only height of the plant is considered, that is only one character is considered. So what is a difference? A monohybrid cross is a genetic cross between homozygous individuals with different alleles for a single gene locus of interest. Okay, so here we consider a homozygous individual. So if you remember, capital T and small t, these are the two homozygous plants. Okay, so these are having different alleles for a single gene. Okay, while in case of dihybrid cross, it is a genetic cross between the individuals with different alleles for two different positions of the gene. Here the genes, they are positioned at the same, single gene is considered. Here the two genes are considered. That's why monohybrid and dihybrid. One character is considered and studied at a time in case of monohybrid. In case of dihybrid, two independent characters are considered and studied. In case of monohybrid, a single pair of gene is involved. In case of dihybrid, two pairs of genes are involved. In case of monohybrid, the predicted phenotypic ratio is 3:1, 3 tall and 1 dwarf plant, if you remember. While in case of dihybrid ratio, the phenotypic ratio is 9:3:3:1. Okay, it is 9:3:3:1. Important determining of the characters, or the important for determining the dominance relationships of the alleles, and this dihybrid, it is important for assessing the independent assortment of the allele. So the difference between monohybrid and dihybrid ratio can be asked in the exam. If it is asked for 2 marks, 4 points you have to write, and if it is asked for 3 marks, you have to write 6 points. You can add one example here, okay, and you can write the points. Okay, so that's all about the basic 3 laws of inheritance proposed by Mendel. Each law can be asked in your exam separately. So study each law with the example. And also the difference between monohybrid and dihybrid cross can be asked. So prepare a chart of the differentiating points, okay? And try to understand the concepts thoroughly, so you will be able to justify your answers completely, and remember all the biological terms with respect to their examples. So hope you have understood today's lecture. Do study, read your PDF book, and keep your notes ready for your studies. And I will be taking your free online tests after each lesson, completion of each lesson. Okay, so that you can examine yourself, and those who want to prepare for the NEET examination will be definitely scoring good uh, by attempting the online tests. Okay, so keep studying and keep doing smart work, and that's all for today's lecture.